Hi guys, Miss Vance again. This is Unit 9 Notes, Part 2, and we're going to talk about the first meiotic division called Meiosis 1. One thing I want to point out to you before we start is that the stages or the phases that occur in meiosis have the same names and the same similar things happen as happens in the stages of mitosis. And so the big difference is that how the chromosomes behave. So pay close attention as we talk about the first division. So in meiosis 1, this is the first division, okay? In prophase 1, it's very, very similar in a lot of ways to prophase of mitosis, okay? It happens in the nucleus. The chromosomes coil and become compact. The nuclear envelope starts to break down and move away. The spindle starts to form. Very, very much like happens in mitosis. Here's a difference, okay? In meiosis 1, prophase 1, the homologous chromosomes come together as pairs. By this process we call synapsis. That means that the duplicated chromosome number 1 from your mom and the duplicated chromosome number 1 from your dad will pair up together, forming something called a tetrad. This is the two duplicated chromosomes of the same type. They have 4 chromatids basically, and they form a tetrad. That doesn't happen in mitosis, but it does always happen in prophase 1 of meiosis. Here's another thing that's different. When they're paired up in the tetrad, the non-sister chromatids exchange genetic material by a process called crossing over. How does this happen? Okay, you've got the chromosome number 1 from your mom and the chromosome number 1 from your dad. And basically, the chromatids that are from one chromatid from dad's chromosome and one from mom's chromosome will cross over each other and trade portions of their chromosomes. It's kind of difficult to describe without seeing a picture. So let's look at a picture here. In interphase, again, interphase looks very, very similar to what you see in mitosis. If you were just looking at the cell, you couldn't really tell whether it was in interphase of mitosis or meiosis unless you knew exactly where the cell came from. Because remember, meiosis only occurs in the sex organs, in the testes and ovaries. We call it, the generic term for testes and ovaries is the gonads. That's the only place it occurs in humans and other animals similar to us. And so you couldn't tell the difference. But then in meiosis 1, you start to see things different. Okay, in prophase 1, you see the tetrads forming. Here we have the 3 different chromosomes, number 1, number 2, number 3, that are lined up together. Dad's chromosome and mom's chromosome lining up together and forming that tetrad. 4 chromatids there. Tetra means 4. And in that process, they're crossing over the non-sister chromatids and exchanging portions of their chromatids with each other. This allows you to pass on a chromosome that has a mixture of traits from your mother and your father to your offspring. All right. In metaphase 1, the tetrads line up at the cell equator. That's something that's different also because, again, the tetrads stay together and they line up at the cell equator. They still attach to spindle fibers. A big difference here is that the spindle fibers from one end attach to one of the chromosomes, and the spindle fibers from the other end attach to the other one in the tetrad. And so when anaphase 1 occurs, this is also different. The homologous pairs separate. Notice that the chromatids don't separate from each other. The chromatids are still attached together. The duplicated chromosomes are separated. But because one of those chromosomes is attached to one end of the cell and the other chromosome is attached to the other end of the cell, that separates the two chromosomes, the 2 homologous chromosomes, from each other. So here we have, notice that, again, dad's chromosome is attached to this end of the cell. Mom's chromosome is attached to this end of the cell in metaphase 1, and then in anaphase 1, when the spindle fibers start pulling the chromatids apart, their chromosomes apart, they're pulling entire duplicated chromosomes from each other. And notice now you've got in those chromosomes, instead of all blue or all red, you've got a mixture of blue and red because of crossing over that happened in prophase 1. In telophase 1, It's very similar to telophase of mitosis. Again, the duplicated chromosomes reach the poles. They start to unwind. In some species, a nuclear envelope reforms around the chromosomes. In other species, not so much. But the big difference here is that each nucleus, rather than having the diploid number of chromosomes, like the original parent cell, has only the haploid number. They have half the number, even though the chromosomes are duplicated chromosomes. You have, if you're counting chromatids, they would have the same number, but the chromatids are still attached together at the centromere, so they're still considered one chromosome. And so in the picture here, we have telophase 1 and cytokinesis. Notice that the cells now have only 3 chromosomes each instead of 6, even though those chromosomes are duplicated. The nuclear envelope starts to form, the cleavage starts to separate the cells into daughter cells, and we have no spindle forming there until the next division. Now, the last thing we're going to do in today's notes is watch a little video. This comes from your textbook from Pearson, and it's a really good video that shows you the entire process. And then in the next set of notes, we'll pick up where we left off and talk about the second meiotic division, meiosis 2.
Humans produce gametes, eggs and sperm, through the process of meiosis. Here, we'll follow the production of male gametes by focusing on this cell as it goes through meiosis. Let's begin in the nucleus, where genetic information is stored in chromosomes. Most of a person's cells are diploid, with 2 sets of chromosomes. One set is from their mother, shown here in red, and the other set is from their father, shown in blue. Each maternal chromosome has a corresponding paternal chromosome. These matched pairs are called homologous chromosomes. During interphase, chromosomes are duplicated. Each chromosome now consists of 2 identical copies called sister chromatids. Zooming in, we see that each sister chromatid is made up of DNA wound around histone proteins. Each strand coils up into a tight helical fiber. As meiosis begins, a spindle forms and duplicated centrosomes start to migrate toward opposite poles of the cell. Back in the nucleus, the chromosomes are condensing. In meiosis, homologous chromosomes stick together in pairs. The close association of homologous chromosomes allows segments of non-sister chromatids to trade places. This recombination of maternal and paternal genetic material is a key feature of meiosis. After the spindle forms and the nuclear envelope breaks down, microtubules from opposite poles attach to each chromosome of the homologous pair, resulting in a tug-of-war. At metaphase 1, the chromosome pairs are positioned in the middle of the cell. The next stage begins when homologous chromosomes separate from each other and move toward opposite poles. Each chromosome still consists of 2 sister chromatids. This cell began meiosis with 46 chromosomes, but each daughter cell now has only 23 chromosomes. In meiosis 2, microtubules from opposite poles attach to the chromosomes, which then move to the center of the cell. Next, the sister chromatids separate, becoming full-fledged chromosomes that move to opposite poles. Nuclear envelopes reform, and each daughter cell divides into 2 cells. We started with a single diploid cell, and now that meiosis is complete, we have 4 haploid cells, cells with a single set of chromosomes. These haploid cells mature into gametes that can contribute to the next generation.